identified were 4 factors, as they called them, and these are now by some people referred to as Yamanaka factors because of his lab having identified these first. Lots of other people did it too. But so these 4 genes that are that they identified in their collection are all genes that are regulating, as scientists have already studied them in other contexts, they're important for regulating gene expression, so turning genes on. And when they put these 4 factors into a fibroblast, they could make an embryonic stem cell. So this one looks a lot like the embryonic stem cells that I showed you earlier in the talk. Just to emphasize this idea of gene expression a little bit more, if you have a somatic cell, sorry, I'll start with the stem cell here. If you have a stem cell, ES cell, it will differentiate into a somatic cell. If you look at the actual genes expressed, and this is just a cartoon representing 3 genes, there are many more, but there are genes like these shown here, NANOG, OCT4. These are genes that are on in embryonic pluripotent cells. But as those cells differentiate into a somatic cell, like a fibroblast, they all get turned off. So these are really clear identifying features that will allow scientists to distinguish between differentiated versus pluripotent cells. So reprogramming, which Yamanaka did here with his 4 factors, allowed these cells to look like stem cells, and then when scientists looked at them, they did see that all the genes that they thought should be there, that were normally found in embryonic stem cells, were turned on. So putting in these 4 factors then initiated and triggered all these genes that are normally found in embryos to be expressed. And that is what then allowed those embryonic, those cells then to take on this embryonic